Welcome back to the Business of Biotech. I'm your host, Ben Comer, Chief Editor at Life Science Leader, and today I'm speaking with Cyril Konto, MD, President, Executive Director, and CEO at Ichnos Glenmark Innovation, or IGI for short, a clinical stage biotech developing multi-specific antibodies and headquartered in New York City. Cyril is an oncologist and has worked in big pharma clinical development at BMS and Pfizer. His team developed elranatamab, a bispecific T-cell engager approved by the FDA in 2023 as Elrexfio. And he also led Allogene's early development of allogeneic CAR T-cell therapies for lymphoma. Cyril became CEO at Ichnos Sciences in 2011 and CEO at IGI in 2024. And I'm excited to hear about the IGI business model, which is unique, and how the company scored a $700 million upfront deal with AbbVie for ISB 2001, a tri-specific T-cell engager for multiple myeloma. We'll also talk about why IGI is focused on bringing innovative therapies to emerging markets and how capital flow in China is changing the way IGI communicates externally. Cyril, thank you so much for being here.
Thank you, Ben. It's a pleasure to be with you today.
I'm glad to have you on. I mentioned in the intro you are an oncologist by training. You've worked at two of the largest pharma companies in the world. How did you first become interested in biologics specifically, and what could you tell me about your past career experiences, either successes or failures that led you to Ichnos Sciences?
I'm an oncologist by training, and early on, I saw firsthand the limitation of traditional therapeutics in complex cancers. Um, Biologics open the door to precision, modularity, and combinatorial biology. The ability to engineer molecules that solve problems, small molecules simply can't. So that's how I got interested. My time at companies like BMS and Pfizer grounded me in the rigor of large-scale R&D while teaching me what slows innovation and really made created the appetite to join the biotech world. Um, my successes, among them, development of ipilimumab, supporting the development of nivolumab, anti-CTLA-4 first, PD-1 second, you mentioned elranatamab, BCMA, CD3 bispecifics. Those successes taught me discipline. The failures, and there were many, taught me humility and the importance of platform thinking. Um, at BMS, we had blinded monoclonal antibodies against many different IO targets. Pfizer, we touch upon many technologies. We build up a portfolio of allogeneic CAR-T through collaborations with Selectis and Servier that we spun off to Allogene, and Allogene is running the registrational trial for those allogeneic CAR-T. So those are the different elements of drug development in biotech world, in pharma at a larger scale. You sometimes win, but we have to face many failures. IGI represented the perfect inflection point, a chance to build a company around multi-specific science with the agility of a biotech and the scale readiness of a global alliance.
That's great. And I want to get into IGI's business model in just a second. But before we could get to that, up to 2024, you were CEO at Ichnos Sciences in 2021, the very beginning, of the biotech winter, a very difficult time that's only starting to thaw out now. I wonder what you could say, about, and I believe this was your first CEO role as well. But I wonder, Cyril, what you could say about how you managed through that period of time, and what the difficult decisions were that you had to make to keep the company alive and growing.
So first, Ichnos Sciences Inception was in late 2019. It was a spin-off of the biologic research unit at Glenmark Pharmaceuticals. It was created with the idea that a biotech would uh, bring the right ecosystem for drug innovation, especially with biologics, in what was back in time a generic-driven pharma company, Glenmark Pharmaceuticals, which remain the sole investor. We are a fully owned subsidiary of Glenmark Pharmaceuticals. We and just to complete the picture on why we're speaking IGI today, um, the good work we've done, and I'm going to respond to your question in a few minutes, made Glenmark decision to allocate their small molecule portfolio to Ichnos Sciences and to make sure that every employee recognize themselves in this new structure. We decided to create a new branding, a new corporate name, Ichnos Glenmark Innovation. So now, how did I keep the company alive during the biotech winter? 2021 required decisive leadership. Capital tightened, science became more competitive, and investors shifted sharply toward de-risked assets. My priority was to focus the pipeline, make evidence-based decision, and allocate capital with discipline. We exited areas that were not strategically aligned or insufficiently differentiated. As example, for areas, inflammation and immunity, we were more experts in cancer. We decided to out-license our portfolio for those assets in this specific area and focus in oncology. When it comes to assets insufficiently differentiated, we had a CD38, CD3 bispecific in a crowded space with standard first generation bispecific T-cell engager. We decided to eliminate this asset. We also were running low in capital. We couldn't spend in too many programs. We doubled down on assets where IGI had a true competitive advantage. Multi-specifics or multi-specific antibodies with validated biology and a pipeline with line of sight to clinical differentiations like ISB 2001, a first-in-class CD38, BCMA, CD3 tri-specific T-cell engager. Survival required both operational austerity and scientific ambition. We executed both. Last, in this spin-off I mentioned earlier, we also had a manufacturing plant allocated to Ichnos Sciences. In this difficult environment, with also the need to make sure that we acquire the ability to scale up, we decided first to divest this asset, which was certainly not the right time in the post-COVID era. It was extremely difficult to find buyers for our manufacturing plant. And without finding the right buyer, we decided to shut down our own manufacturing plant and tech transfer our technologies to reputable, large-scale commercial CDMOs to enable the next phase of development for Ichnos Glenmark Innovation.
And that was an antibody manufacturing center or small molecule?
A biologics
Okay.
manufacturing center where we did cell line development, manufacturing, operations, analytics, and also QA/QC.
So it was actually more affordable then to close the internal manufacturing and transfer over to a CDMO as opposed to running it yourself. That's interesting.
More affordable, more flexible. Realize that we are an emerging biotech company, those are molecules that we've administered first to patients, we don't know whether they're going to be drug products, marketable drug products. So there were substantial operational expenditures allocated to manufacturing those molecules without really knowing whether one day one of them would cross the finish line of marketing approval. So it's also a lot of investment, and by shifting from in-house manufacturing to an outsourced CDMO model, we were also complying to the risk model of biotech company in general.
Right. And multi-specifics is a term that has been trademarked by Ichnos or by IGI, which is fascinating. I'll let you tell the story of that. But was in terms of finding a CDMO, transferring, that process over to a third party, did you find that there were plenty of CDMOs out there that could handle, that particular type of manufacturing process, or was it difficult to find a partner that could essentially, do what you wanted them to do?
I say the skills were there. We had all we discussed with mid-sized CDMOs, mostly, not large CDMOs. But the risks in this post-COVID, where a lot of CDMO had started to produce vaccine and biologics,
Right.
were
So
there.
capacity issues.
Indeed, we had to make some level of commitment to purchase our supplies from the buyer, a significant commitment over multiple years to make sure that the buyers can also on its own grow, find new customers, replacing progressively our own purchases. So there was also a financial commitment and very few capital to expect from selling a CDMO. So the environment was overall less permissive than switching directly to a large-scale CDMO like Thermo Fisher or Catalent.
Right. Getting back to IGI, I want to ask you about the business model. You mentioned, IGI is formed through an alliance between Ichnos Sciences, which was originally a spinoff from Glenmark Pharmaceuticals, which was traditionally a generics manufacturer. What more can you say, Cyril, about, why maybe you didn't just stay as Ichnos Sciences? Why did you then need to create the Ichnos Glenmark Innovation company that's separate and that is now headquartered in New York City? What was the thinking behind that particular model?
So IGI was intentionally designed as a focused, innovation-driven biotech with the scale advantages of global pharmaceutical company, Big Brother Glenmark Pharmaceuticals being backing us up. Glenmark carved out its biologics unit to create IGI, a company with its own governance, P&L, R&D autonomy, while still benefiting from Glenmark's infrastructure and global reach. This model gave IGI the space to innovate boldly while ensuring stability during economic turbulence. So that's really, to my mind, what supported every scientist, every clinician, supportive function in IGI to focus on innovation, drive innovation through research labs to clinic. Why did we shift it from Ichnos to IGI? I think this is more making sure that we have a cultural harmonization.
Hmm.
Everyone in every Ichno genius can recognize themselves in the IGI. We are headquartered in New York. We're doing research in Lausanne, Switzerland, plus some manufacturing oversight in Neuchâtel, Switzerland, and we have a supportive R&D hub in Mumbai, India. So you see, we're we're a multi-location firm, hence a need to harmonize culture. We have different way of thinking, of interacting, of formulating comments. US is very entrepreneurial. Europe is more reserved when it comes to comment and criticize others. India is very respectful of hierarchy. So we had to create a common denominator, excuse me, that is our multicultural.
From a people perspective, Cyril, how do you bridge those gaps between three, very different regions culturally, and you as the leader at IGI, what can you say about how you bring those very disparate groups together and under one mission?
I think our greatest achievement is our culture. We created scientific rigor, transparency, and the courage to make hard choices. We've learned to pivot when needed, terminate what doesn't deliver, and invest where data lead us. And with that, and the importance of repeating this at multiple occasions every quarter, we're broadcasting a town hall from the New York office where all the other locations are connected, and realize that we have 9.5 hour time zone difference from the earliest to the latest time zone in our organization. So everyone, every single Ichnos in my organization is also committed to connect, attend those town halls where we repeat our strategy, where we communicate our achievements, where we are learning also through some of the failures that every biotech has to face. So all of this is a support to a great culture that makes IGI so special today.
The top of your investor presentation that I looked at when I was just looking through your website, it says collaboration propels innovation at the very top, and I sort of related to what you've just been saying. What would you say about IGI's mission, collaboration, really focus on collaboration? How does that differ from maybe some of the previous companies that you've worked at? And is there a clear advantage to this focus on collaboration?
Our tagline, as you said, is collaboration propels innovation, and we feel that alone we will not be able to deliver a high-scale innovation relative to having a biotech to biotech collaboration, biotech to big pharma collaboration, going back to the AbbVie deal you mentioned in the intro, or even biotech to academia collaboration. I'd say IGI works differently because collaboration isn't a slogan. It's an operating model. It's in our DNA. I took the helm of Ichnos Sciences at the beginning of 2021. We signed our first licensing agreement with Almirall, a Spanish dermatology company, to develop our anti-IL-1 RAP monoclonal antibody for hidradenitis suppurativa, a derm disease. They are the specialist. Going back to my prior comments on focusing in cancer and out licensing our assets to other experts. So that was in December of 2021. And we all just announced that this asset is now moving to phase 2.
And that was
Um,
discovered internally.
yeah, it's our internal research. We deliver. We had an IND ready asset. Almirall ran the phase 1 first in human trial. We even had proposed a draft protocol of this for this study. 2 years later, we out-licensed our anti-OX40 monoclonal antibody to Astria Therapeutics. a company specialized in rare disease and dermatology. And they've run the dose escalation in healthy volunteers for our monoclonal antibodies. They also enhanced the antibody we delivered to them with a YTE half-life extension technology to make this asset a best-in-class. So 2 examples of experts we decided to partner with. Our scientists, clinicians, translational teams, and external partners engage early and often. We have those joint steering committee. With AbbVie, we went even beyond the standard joint steering committee. We created subcommittees to enhance the collaboration. In contrast to Big Pharma, we run a flat, integrated R&D environment where decisions are fast and evidence-driven. The advantage is speed, clarity, and accountability, and our partners love that from us. The keys to collaboration are transparent goals, early scientific alignment, data discipline, respect for each function's expertise, and cross-functional ownership. So that's what defines our operating model. We encourage everyone in IGI to put themselves into the shoes of our partners. Partners have sometimes different priorities, different imperatives than the one we have foreseen for our assets, and projecting ourselves into our partner on the partner side is also helping understanding and speeding up clinical development, something we've done extremely well with Astria, with AbbVie, and Almirall.
Yes, you have certainly done very well with the collaborations, and I want to get back to ISB 2001, a tri-specific antibody targeting multiple myeloma. AbbVie last summer licensed that for $700 million upfront. And you mentioned already, the two other deals that IGI has done for a company that has not been around for very long. That's quite a solid performance. I wonder if you could talk a little bit maybe about the AbbVie deal in particular. How did you make that deal? And also, what does it mean for IGI from a financial standpoint?
So we started the first in human trial with ISB 2001 in November of 2023. We designed a dose escalation sufficiently agile to move quickly to top doses, while also preserving a limited number of patients treated at suboptimal therapeutic doses. That's why we get in December of 2024 an oral presentation at the American Society of Hematology, where we presented preliminary results of the first 12 subjects treated with ISB 2001, showing early clinical efficacy at low doses, as low as 50 microgram per kg. So it became for us critical to make sure that we put ISB 2001 into a BD track to find a partner who can accelerate clinical development and enable optimized commercialization for this asset. With our ambition again and strong hope behind this asset with the limited data we had back in time. So for ISB 2001, all the big pharma players in myeloma came to our data room, different stages of our bidding process. We, IGI, did a thorough strategic fit assessment. They were not all the same for us. Some had bispecific already in their pipeline. One has a trispecific in its pipeline. heavily involved in myeloma. We were a little bit scared about this one. We didn't want our asset to land in the hands of a big pharma, just like shutting the program down to protect its own portfolio. We created significant FOMO with all those key players in the data room. But that was not enough. We also put in place an external publication plan, making sure that we would receive either good news in terms of regulatory achievement or good news in terms of podium presentation during the bidding process, and especially during the negotiation phase of this bidding process that lasted from December to December 2024 to July 2025 with the execution of the deal with AbbVie. Those regulatory milestones, we received fast track designation for ISB 2001 in March of this year, when companies were sending us their term sheets, and we would only open our data room to serious companies with significant terms in the term sheet to protect ourselves, because we're small and we cannot address questions from too many big pharmas. We wanted to have meaningful and high quality responses to serious bidders, and also to protect the confidentiality of our data. We didn't want to let anyone get into the data room, get access to our data, and then leave without being really serious on the licensing of this molecule. And the last thing is, in January, we filed the abstract to ASCO. We never thought that we could get an oral presentation at ASCO, but here we were helped by some luck. This year, ASCO started a rapid oral presentation for drugs like ISB 2001, and we were lucky enough to be granted a podium presentation for ISB 2001 next to our direct competitor, J&J 5322, Janssen trispecific targeting BCMA, GPRC5D, and CD3, which would help also our bidders to compare and contrast the two trispecific in development for myeloma. And lastly, when it comes to this deal, it's very important to receive the right counsel. So we haven't been cheap in selecting our legal firm. We picked Covington and have been extremely satisfied with the deep service we received from them. The bills were to the extent of the work done, definitely, but this really helped because as a small biotech dealing with big pharmas, you can expect that on the other side of the table, we had the big firms. And when it comes to other partners, there were the Latam and Walkins that are very famous as well. So that's also helped us anticipate all the scenarios of the life cycle management of this molecule in our licensing agreement, and at the same time protect the emerging market rights that we wanted to keep for ourselves and license out to Glenmark Pharmaceuticals, our parent company, with the prospect to become the leader of therapeutic innovation in emerging markets. So that's also guided us in our choices.
You made several, I think, really important points just then, Cyril. Protecting your communication and data to make sure that that's not getting into the wrong hands. You anticipated a potential catch and kill and avoided that. You mentioned FOMO, fear of missing out. That podium presentation, I suspect, really helped build up that FOMO. And then you mentioned the emerging markets focus too, which I think is also very interesting. The out licensing deal, I believe, with AbbVie covers the US and Europe, Japan and China. Am I right about that? But you still have the rights to other emerging markets. Can you talk a little bit more about I guess, Glenmark's role in potentially commercializing those and why you wanted to maintain those emerging market rights and what the opportunity is there.
You're absolutely right. We granted AbbVie exclusive licensing rights for ISB 2001 in North America, Europe, Japan, and Greater China, retaining the rest of the world territories right in IGI for subsequently licensing those rights to Glenmark Pharmaceuticals. Um, it's it was also bold from AbbVie to accept this type of deal. But what we did within IGI is making sure that we were protecting AbbVie's territory and commercialization, more importantly. When you think of global revenues, AbbVie's territories represent roughly 85 to 90% of the sales, and emerging markets the remaining. So it's also in the interest of IGI to make sure that AbbVie can develop ISB 2001 fast, efficiently, and get this product to market as soon as possible. Not mentioning the $1.2 billion milestone payments plus the royalties on net sales that IGI will benefit from this strategy. So we also created within our licensing agreement mechanism to protect AbbVie in their development and commercialization plans for not for preventing them from being affected by some wrong moves that could happen in the emerging markets. And that is until commercialization in the US. So that's also when ourselves projected into AbbVie's position on what could make the partner less excited by striking a deal with IGI given this context. That being said, we also believe that Glenmark Pharmaceuticals operations footprint in emerging market is large, at scale, and probably the best fit for developing eventually out of what AbbVie is doing in the Western world by creating some bridging studies to match local requirements in emerging market, and more importantly for commercializing ISB 2001 in those emerging markets. So from an IGI standpoint, we have the best of both worlds taking care of ISB 2001.
So, just so I'm clear, in order to avoid, any potential missteps in emerging markets, those bridging studies, other development work in the emerging markets that will be conducted by IGI or Glenmark, those will happen only after AbbVie receives approval and begins to commercialize in the West. Is that right?
Not necessarily after, but if it happens before, it will be under AbbVie scrutiny, and nothing will be done to harm AbbVie's ability in their development and commercialization.
Right, got it. The other, I think, important point that you made previously that I didn't mention in my recap is the importance of contracting and having, a strong partner who can help out a small biotech to match the kind of firepower that big pharma would have. That's incredibly important as well. I didn't want to leave that out. IGI, we've talked about this, is focused on multi-specific antibodies. Your background is in biologics, but you also have a small molecule candidate. What can you say, Cyril, about your development strategy across those two very different modalities?
While our core strength is multi-specific antibody, biology does not respect modality boundaries. Some targets are best addressed with protein engineering, others with small molecule. Our strategy is to deploy the right tool for the right biology. not force biology into a single modality. This dual modality approach allows IGI to build a pipeline that is both scientifically coherent and commercially diversified. We have inherited a single small molecule candidate from Glenmark Pharmaceuticals. The code name is GRC65327. It's on our pipeline slide on the website. This is a CBLB inhibitor. I'm not going to make a case for the CBLB pathway. In essence, CBLB inhibition supercharges the immune synapse by enhancing activation of T cells and NK cells. So when you think of this biologics biotech company developing multi-specifics, engaging T cells and NK cells, there is an opportunity for combination by enhancing the effector cells responsible for tumor killing. Of course, all of this will be upon confirmation of the safety of GRC65327, which is planned to enter clinic at the end of 2026.
Okay. I wanted to ask, kind of following on this question about multispecifics, you have what's called the BEAT platform at IGI, and I wonder if you could just describe what that is, if it's possible at a high level, Cyril, and kind of which I don't know if that's possible or not. We're not a scientific podcast, so I won't ask you to go, or I'll ask you to stay out of getting too far into the weeds. But can you at least give us a sense of what makes the Beat platform unique?
Uh, BEAT is IGI's proprietary multi-specific engineering platform. Without getting into too much protein details, I'd say it enables precise modular assembly of antibodies with multiple arms. When you think of ISB 2001, there are three arms and three different arms, whereas a standard monoclonal antibody has only two arms and they are similar arms. So that's different specificities, different effector functions as well. It solves long-standing challenges such as chain pairing, geometry, and stability for multispecifics. BITS allows us to design molecules that engage multiple tumor antigens, selectively activate T cells, NK cells, myeloid cells, so a broad spectrum of immune cells, avoid off-tumor toxicity. That's very important because some of those targets can be expressed on the tumor, but also on healthy tissues, and deliver stronger immune synapse formation. So, in short, it's more biology in one molecule engineered for real world clinical impact.
There has obviously been some success already with bispecific antibodies. You're now working on trispecific. I mean, why do you think that, there I guess is the next thing going to be, quad specific, or are we going to keep adding, additional specifics to the antibody? Why, I guess first tell me, why there was a need to move from bispecific to trispecific, and then if you have any thoughts about, what comes next after that.
And we're at the core of the purpose of our BIT multi-specific platform. Yes, you're right. There was a need. The tumors are extremely smart. They don't regulate antigens to avoid attack. So by having multiple binders targeting multiple antigens on the tumors, we avoid the main mechanism of resistance associated with antigen downregulation. The multi-specific can also activate more deeply the immune cells. You can drag a T cell, but you can also enhance the activation of these T cells. You can hit NK cells on multiple activation pathways, which is critical to the NK cell biology. So those are the benefits of adding additional binders, creating this multi-specificity on the BIT platform. That being said, there is a biological challenge. We create those molecules with multiple binders. We have to make sure that they do not bind to everything but the tumor because of those multiple arms. We even invented this term in-house of pentopus. We have a platform that can deliver multi-specificity beyond tri-specific. And I'm glad to report that we just nominated our first NK cell engager in our what we call Immunite platform of NK cell engager because we believe in the immune power of NK cells to kill solid tumors. And we expect to bring this ISB 2301 NK cell, multi-specific NK cell engager into clinic in early 2027. This will be our first example of delivering a molecule beyond the standard tri-specific. The BIT enables this because the BIT is stable. So far with ISB 2001, we're observing an half-life of more than 2 weeks in patients. It's close to the normal half-life of monoclonal antibodies, despite having this highly complex technology. So that the bit stability is a key component to the multi-specificity strategy we're developing in IGI.
When you start adding binders and going into more and more, from bi to tri-specific and even more multi-specific type of molecules, there's obviously, we hear a lot about personalized medicine in oncology and, creating almost individualized therapies for patients that are specific to their cancer types. When you start adding these additional binders and get into the multi-specifics, are you also bringing down the size of the patient population that those drugs would potentially target? Are they getting more personalized, or is that not how it works? Is that not correct?
It's more personalized to certain solid tumor indications when it comes to ISB 2301. It's dedicated to myeloma, smoldering myeloma, and eventually some autoimmune indication for ISB 2001 because we designed the molecule to specifically target the plasma cells that are the problem of myeloma or autoimmune disease. So in some way, we are creating a technology that is specialized and which hopefully will deliver enhanced anti-tumor activity due to the over-specialization we brought to the technology against solid tumor indications. That being said, some very dedicated technologies targeting one antigen requires to test patients for this antigen. Best example is Herceptin. We had to test patients for HER2, making sure that they had overexpression of HER2 and those patients were the right candidates to receive Herceptin. With a multi-specificity, if you're targeting multiple antigens, you have less of a need to check on the marker expression because we're creating this binding of multiple antigens and the biology makes those antigens more or less expressed, but this is compensated by the multi-specificity binding to different tumor antigens. So it's kind of balanced, but you're right. We're designing molecule for specific tumor indications, not like chemotherapies with a very broad impact.
Right. Excellent. A lot of capital, it's a big topic in the industry right now. A lot of capital is flowing into China and Chinese biotechs. It's something that I heard FDA Commissioner Macri mention recently from a kind of speed and competition standpoint. He's obviously got a number of pilots going on at the agency to try to bring drugs to market faster. But I wonder if that trend of capital moving into China has impacted IGI's strategy in any way.
It it did, yeah. And you're right. I think Chinese biopharma industry has come a long way. The biotech sector is scaling rapidly, backed by capital, regulatory speed, large pools of patients. There's a number of biotech company listed in the Hong Kong or Shanghai exchange that has grown. It's five times between 2018 and 2025. China's growing influence also in global biopharma innovation. They file seven times more INDs than the US today, and almost twice the number of NDAs than the US
It's
today. So
amazing. Yeah.
indeed, it's very hard to compete. So the dynamics affects global timelines and partnering expectations. Our parent company has announced multiple in-licensing deals from settled with Chinese biotech companies. Angry being the last one. So it's for us. Uh, it's important to put this into consideration. Uh, How can we differentiate our innovation from what China is doing? And China has clearly evolved into innovation-driven biotech sector. Um, we believe that the market competition reinforces our need to stay ahead through quality science, differentiated platforms, and selective geographic partnerships. How did this affect us? First, a major flow of capital, US capital to China, so less capital for biotech. Hence, a need to focus on non-dilutive capital through partnership deals like the one we struck with AbbVie. Second, we we are trying to avoid to feed Chinese innovation with our internal work. And the best example, I think, was striking to me to see the preclinical data package of our main competitor, Janssen Trispecific, published only recently, after about 200 patients have been treated in clinic, and the asset is already on a registrational path. So giving you enough leeway to get closer to BLA approval before informing the competition on the design of your molecule and the preclinical performance of your molecules. That's, I think, this new late peer review publication model is a model that we will implement in IGI, especially when it comes to ISB 2301. And last is everything is about intellectual property. We're filing our provisional certainly sooner than what big pharmas are used to do, because we also have a need to communicate on our technology. We will seek licensing opportunities for ISB 2301 ex-US. We intend to keep the rights for this molecule in the US, not only development, but also commercial rights for ISB 2301 in the US, and eventually, upon the success of this asset, transform our organization from an R&D biotech today to a commercial biotech in a 6 to 7 years horizon. So that's how we're being influenced. But I think I think we have to also think of ways of modernizing the way we do drug development. today. The time to first patient dosed is a critical time. Can we enhance reviews at US clinical investigational sites in parallel of the FDA? Can we make sure that startup activities in the clinical operations space can also start before the IND clear, the FDA clears our IND? So those are ways of modernizing drug development in the Western world. There's also a significant cost of drug development compared to China. We have to acknowledge that we have a different healthcare system model than China, and it is more expensive to do drug development in the US, in Europe, and in Australia than it is in China. And I realize why some of my peers in the biotech sector are interested in driving innovation in China or getting innovation from China.
To your point on the need for modernizing the FDA, another, initiative that Commissioner Macri has been talking about, and when I saw him speak a week or two ago, said that, very soon he would be announcing specific either disease areas or modality types that would no longer require animal studies in preclinical research. And I wonder, what you think of that, Cyril. How has the technology in areas like in silico development reached a point that animal studies can be avoided in certain areas? What's your experience with that, and what are your thoughts on getting rid of animal studies, at least in certain modalities or in certain disease areas?
IGI remains compliant to all health authority regulations, and we applaud the Animal Welfare Act that the FDA is enforcing whenever it is possible. I say from our standpoint, and think of ISB 2001 first, one of our binders did not cross-react to cynos, to cynomolgus monkeys. And because this molecule cannot bind to some specific cells it's supposed to bind in human, in monkeys, those animal models become irrelevant. So we had discussion with the FDA. We presented the molecule, we presented the preclinical data package, and we also presented our in silico PK/PD modeling in order to predict a safe dose range, but also an efficacious dose range through computed modeling. And the FDA really welcomes this opportunity. We partnered with a key player in the field of quantitative systemic pharmacology, Certara. We built this model, and interestingly, our clinical data match pretty well the predicted data by this in silico model. And you can already speak of AI here. That's some of the ramification of artificial intelligence. And we have a manuscript. We submitted our QSP manuscript to explain the world how we thought through this exercise. So that was for ISB 2001 at a small technology scale in the very large scale that IGI aims at. Now think of ISB 2301. We have even more than 3 binders without disclosing too much. Then we are reaching such a level of biologics complexities that it's very hard to recapitulate the mode of action of this molecule in animal models. Of course, you multiply the binder, so you multiply the risk that binders will not bind animal models in the right way, and that the data you're going to collect in monkeys or other species will not be relevant to do pharmacokinetics or even non-clinical toxicology relevant experiments. So for us, it becomes even more important to partner with those companies and continue our endeavor in silico / AI pharmacokinetics, pharmacodynamics modeling of what our complex technologies will deliver in patients in terms of prediction models.
Yeah, and it strikes me as really powerful to be able to validate the in silico data in your actual clinical trials later in the clinical data later on, which will only make those models stronger over time. That's excellent, Cyril. We're coming up to the end of our time. I'd like to just end with your kind of top priorities right now, your next milestones at IGI. What are you really focused on right now?
We have kicked off our collaboration with AbbVie. We're focused on completing the Trignite phase 1 study. Dose expansion is enrolling fast. We're aiming at providing a recommended phase 2 dose for monotherapy, but also for combination in myeloma. We are enabling AbbVie is taking over clinical development of ISB 2001, and we will meet with health authorities to validate AbbVie's proposal for clinical development plan. So that's for us a major priority. Obviously, we just nominated our first NK cell multi-specific antibody, ISB 2301. We are now working hard on IND-enabling studies to file the IND before the end of 2026 to enable a clinical start in early 2027. That's our second priority. We have enough cash runway to go through the phase 1 of ISB 2301, but we also have to turn to the future. We'll need to prepare for fundraising, and we have to transform IGI from a private company today to a public market ready biotech company. So our third priority is the transformation of our organization. We've done already significant efforts to show future potential investors that we are robust, compliant, disciplined organization. We will continue to do so to enable IPO in few years in the horizon of fundraising for IGI.
Cyril, thanks so much for coming on the show. I really appreciate it.
Thank you, Ben. It was a pleasure sharing ideas and also realizing that what I say can be put into a much larger biotech context. And I'm hopeful that this tiny experience can help my colleagues in the biotech sector, and look forward to connecting with as many of my peers in this to make US biotechnology sector a success.
Absolutely. We've been speaking with Cyril Konto, MD, President, Executive Director, and CEO at Ichnos Glenmark Innovation, or IGI. I'm Ben Comer, and you've just listened to the Business of Biotech. Find us and subscribe anywhere you listen to podcasts, and be sure to check out new weekly videocasts of these conversations every Monday under the Business of Biotech tab at lifescienceleader.com. We'll see you next week, and thanks as always for listening.